In this video, we are going to talk about eukaryotes. So far this semester, we have talked about acellular entities, which includes viruses, prions, and viroids. Remember that acellular means without cells. So these are technically not living organisms, but they do have some of the characteristics of living organisms. We have also talked about prokaryotes, which included the bacteria and archaea. And the prokaryotes are the kind of the simpler cells. They tend to be single-celled organisms, and they don't have the same membrane-bound organelles as eukaryotes, and that includes not having a membrane-bound nucleus. And so now we're going to start talking about eukaryotes. And the eukaryotes, we're going to be talking about them for really the rest of the semester. And we're going to be looking at kind of 4 different kingdoms of the eukaryotes. So we're going to look at protists, fungi, plants, and animals. So first, let's take a look at the characteristics of eukaryotes. So when we talked about prokaryotes in a previous video, we talked about kind of the differences between prokaryotes and eukaryotes. So now we're focusing on the eukaryotes, which are more complex cells than the prokaryotes. And so the kind of the defining characteristics of eukaryotes or eukaryotic cells are that they have a nucleus that is surrounded by a membrane. And this membrane is really the important point because remember that prokaryotes do have DNA in a nucleoid region in the cytoplasm, but they don't actually have a membrane surrounding the DNA. And this membrane is similar in structure to the cell membrane. So it is a type of plasma membrane. Eukaryotic cells also have a cytoskeleton, and so cyto means cell, and skeleton, think of like the skeleton of your body. So these are essentially protein fibers that are found inside the cell. And more specifically, these protein fibers are made up of actin, which is just a type of protein, and microfilaments. And the main functions of these, they do help to give the cell some shape. They help to anchor organelles in place. They may allow for the movement of organelles. And they may be involved in the movement of the cell as a whole. Eukaryotes also reproduce asexually through mitosis. And this is essentially how a cell makes another identical cell of itself. And it's kind of similar to binary fission in prokaryotes, but because the nuclear membrane is involved, that makes it different. So there's more that has to happen with regards to the nuclear membrane breaking down and then being built back up again. Eukaryotes also undergo sexual reproduction through this process of meiosis. There are some additional characteristics of eukaryotes that might not be in all eukaryotic cells, but it might be in several or many of them. So most or many eukaryotes have mitochondria and other membrane-bound organelles. And mitochondria are also membrane-bound. We just like to highlight the mitochondria because they serve an especially important role in eukaryotes. And we'll talk about that a little bit more later. But these other membrane-bound organelles might include things like the endoplasmic reticulum, the Golgi apparatus, things like vesicles, and vacuoles, etc., etc. Most eukaryotes also have linear or bar-shaped chromosomes, and these chromosomes are organized by these histone proteins. So essentially, these chromosomes, so this is a chromosome here. Remember that chromosomes are made up of this double helix of DNA. And that DNA ends up getting wrapped around these histone proteins. And this is essentially a way to organize this DNA. Because remember that these strands of DNA can be very, very long. And if they weren't organized, they could get tangled up. And essentially, the genes would not be accessible for transcription, translation, protein synthesis, gene expression. And so these linear chromosomes are actually made up of these histone proteins with the DNA wrapped around them. Many eukaryotes also have cell walls, so we're going to see that especially in things like plants and fungi. And then some of them, a lot of them have flagella and cilia, and we'll talk about those more, especially when we're talking about protists and some additional eukaryotic cells. And generally speaking, flagella and cilia are going to be involved in motility, or the ability to move. The evolution of eukaryotes includes this theory known as endosymbiosis, or the endosymbiotic theory. And this cell in the next several slides, are referring to primary endosymbiosis. So we'll talk about secondary endosymbiosis towards the end of this video. So some of the most important organelles that you find in a eukaryotic cell include the mitochondria, which are involved in aerobic cellular respiration. Remember, this is how cells make a lot of ATP by breaking down glucose and other organic nutrients. And then some eukaryotic cells, so not all, but some have plastids, and the most well-known one and the most prevalent one are the chloroplasts. And so the endosymbiotic theory states that these two types of organelles actually evolved when a nucleated cell engulfed a prokaryotic cell. So essentially, there was a cell with a nucleus, so kind of like an early eukaryotic cell. So this would be the cell membrane. This would be the membrane-bound nucleus. And then there was a prokaryotic cell. That became engulfed by this kind of early eukaryotic cell. So essentially what happened is that this cell membrane kind of pinches in, and that prokaryotic cell gets kind of sucked into the host cell. And so what you end up with, and this is happening by endocytosis. So what you end up with is a bacterium that has been engulfed into this host cell that is wrapped around the original cell's cell membrane. And then, of course, we have our nucleus still hanging out here as well. Now, oftentimes when one cell engulfs another cell, it actually kills or breaks down the other cell. But in this case, due to some evolutionary pressure, the engulfed cell actually survives and becomes part of the cell that engulfed it. And in this scenario, both cells benefit. So that's the symbiotic relationship, in that the engulfed cell provides some sort of advantage to the bigger cell. And the bigger cell provides an advantage to the smaller cell. And then over time, these two cells become so integrated with each other that if you were to then try to separate them, they're not able to survive on their own. So, for example, if this is a modern day eukaryotic cell down here, if we were to take, let's say, this yellow thing, let's just say that we're talking about the mitochondria. If we were to pull the mitochondria out of the eukaryotic cell, the mitochondria would not be able to survive, and the eukaryotic cell would not be able to survive. They would both die because they need each other in order to survive. Now, there are several bits of evidence that support this endosymbiotic theory. So first of all, mitochondria and chloroplasts are similar in size to bacteria. They also have their own circular DNA. Basically, their genome exists in this circular DNA, and they have their own genes. And remember that prokaryotes also have circular chromosomes. The main genome in a eukaryotic cell, so what we find in the nucleus, are going to be those bar-shaped chromosomes, but mitochondria and chloroplasts have the circular chromosomes. Now, some of the genes that were likely in the original prokaryote have migrated to the nucleus, and this is likely what has caused that kind of interconnectedness. That has resulted in the structures not being able to live on their own anymore, because some of the genes from the original prokaryote are now incorporated into the genome in the nucleus of the eukaryote. And so without those genes, the mitochondria or the chloroplast can't survive on their own. Mitochondria and chloroplasts also have their own ribosomes. And remember, ribosomes are involved in making proteins. So this process of protein synthesis. And much of the proteins that they make kind of link back to these genes that they are carrying in their own genomes, so independent of the main eukaryotic cell genome. Mitochondria and chloroplasts also reproduce independently of the rest of the cell, and they do so by a process that is very similar to binary fission, which once again is how prokaryotes reproduce. Mitochondria and chloroplasts are also produced from existing organelles, meaning that the cell, a eukaryotic cell, cannot take lipids and proteins and carbohydrates and nucleic acids and put them together to form a mitochondrion. So cells can do that to form endoplasmic reticulum, to form new cell membranes, to form, new Golgi apparatus. They can, take these kind of independent molecules and stick them together to form these organelles. But mitochondria and chloroplasts cannot be made that way. Instead, you inherit, or the cells inherit these structures from their original parent cell. And then when a cell divides in mitosis and meiosis, the mitochondria and chloroplasts, they do their own binary fission. They do their own replication. So mitochondria can only be produced from other mitochondria, and chloroplasts can only be produced from other chloroplasts. And this kind of goes along with the cell theory. Remember that one of the statements of the cell theory is that cells can only arise from other cells. So cells can't just form. I mean, they did originally when they originally evolved, but since then, all existing cells have to come from pre-existing cells. All new cells have to come from existing cells. And that's the same thing with mitochondria and chloroplasts. We can't make new ones, we just can make copies of what we already have. Mitochondria and chloroplasts also have two membranes, and likely the outer membrane came from the engulfing cell. then the inner membrane came from the prokaryote that was engulfed. So this would be, if let's say this is a mitochondria, this would be the outer membrane that came from the engulfing cell that kind of pinched off. And it's more similar to a eukaryotic plasma membrane. And then the inner membrane is from the prokaryote that was engulfed, and it's more prokaryotic-like in structure. And you can see that on these pictures down here. So with the mitochondria, which is what we have here, the outer membrane is this kind of orangey area. And the inner membrane is kind of this like yellow part in here. And it's folded into these cristae in order to increase surface area. And prokaryotes often have a infolded membrane like that. If we look over here at the chloroplast, and chloroplasts are interesting because they actually have 3 levels of membrane. So they have this outer membrane here, which likely came from the engulfing cell. And then this inner membrane, which could have come potentially from the engulfed prokaryote. And then these thylakoids, these little stacks here, also have a membrane surrounding them. And it's believed that the mitochondria arose from aerobic bacteria known as alpha proteobacteria. And the chloroplast came from cyanobacteria. And cyanobacteria have chlorophyll. And they can undergo photosynthesis. Also remember that some bacteria have all bacteria have a cell wall, and the cell wall has this molecule known as peptidoglycan. And some chloroplasts actually have remnants of this cell wall. So if you have the eukaryotic cell, the outer membrane, and the inner membrane, there might also be remnants of this peptidoglycan cell wall visible in some chloroplasts.
This diagram kind of summarizes the endosymbiotic theory.
So remember we had this kind of initial early cell that then engulfed a bacterium. So in this case, we are talking about probably the alpha proteobacteria that were engulfed in order to make the mitochondria. And with some cells, that's where the story ends. They have mitochondria, but not all eukaryotic cells have chloroplast. But some of these eukaryotic cells that had the mitochondria also went through a secondary endosymbiotic event in which the cyanobacteria, in this case it just says photosynthetic, but we know that it's cyanobacteria, become engulfed in order to make the chloroplast. So when we talk about protists, we're going to talk about some of the algae, which are protists, and some of them became photosynthetic by engulfing other algae. So if we look at this here, so this would be our primary endosymbiosis happening here, where we have, in this case, we're looking at a chloroplast, which was produced by this eukaryotic cell engulfing the cyanobacterium. Remember, this is our primary endosymbiosis, and here we can see that this is our chloroplast. And so this is a eukaryotic cell right here. So it has already engulfed the mitochondria, engulfed the bacteria to make the mitochondria and chloroplast. And then you can see that this whole structure here becomes engulfed by yet another cell. And so in this case, we have additional membranes. So now we have multiple membranes present, which is the evidence for the secondary endosymbiosis. So for primary endosymbiosis, we have this presence of like 2 membranes. For secondary, there are 3 membranes. And I just realized that, this purple spot is not showing the mitochondria. That's actually showing the membrane-bound nucleus. So that's supposed to be the nucleus right there. And that's showing here. So here, this cell has a nucleus, but then the plastid has a vestigial nucleus. So essentially the remnants of a nucleus.